Thank you for the nice introduction. And hello, my name is Byung Tae Lee, a PhD student advised by Professor Hyunwoo Kim at KAIST. First of all, I want to appreciate the conference organizers for providing this great opportunity to share my ideas. And the topic I'm going to present today is about extracting one of valuable information for understanding metabolic networks of an organism, which is called gene-protein reaction associations by using some language models. Gene-protein reaction association explains the connective relationship between the genes and reactions which are catalyzed by enzymatic proteins, which the genes are encoding. Depending on the nature of proteins and reactions, some reactions can be catalyzed by multiple genes, where all of the genes are necessary for catalyzing the reaction, or some of them are just enough, and some of them can substitute the others. And this relationship is often represented by the logical expression using the AND or logic gate. And for characterizing the metabolic network of a cell, this information, the GPR association, allows us to identify the genes which mediate some certain reactions. Finally, we can find target genes. We can find the target genes to engineer when we want to engineer some specific metabolism of the cell to develop the cell into the improved one. So far, 3 approaches are available for finding or assigning the GPR association. One is using sequence alignment to assign the similar forms of GPR associations to genes with high sequence similarity. And the second one is extracting information from the published papers. And the third one is collecting the information from databases which have accumulated the relevant data up to a specific time point. Each method has its own pros and cons, like the sequence alignment method can be the fastest way, but the result is not verified experimentally, and the literature, the information retrieved from the literature can be validated experimentally and up-to-date data, but manually searching the massive amount of data should take a lot of time. And the databases are storing relevant data that are well structured and verified, and they are providing some APIs for convenient data retrieval, but they have a limitation of taking a considerable amount of time to update the latest information. In this context, we have explored new approaches to overcome the limitations of current strategies. Recently, language models based on deep learning technique have experienced huge advances, demonstrated by the outstanding performances in various natural language processing tasks. Particularly, there have been active researches in customizing the language models to specific fields by training the models with the words and context that are related to specific fields. This trend is also increasing in biomedical fields, so that several customized models for the biomedical fields have become available for us. Therefore, we plan to develop a new methodology to extract the GPR association by extracting the information from the literature by using the language models. The information retrieved from the published papers are up to date and experimentally validated. And we can operate this system automatically once we train and optimize the model for our objective. In other words, the goal of this study is to reconstruct the gene-protein reaction association automatically through natural language processing tasks performed by deep learning-based natural language models. To achieve this goal, two natural language processing tasks are performed, and 3 representative biomedical language models are adapted called BioBERT, PubMedBERT, and BioGPT. Each model was optimized for each task, and we established the system by the optimization, and let's take a deeper look at each task. Named entity recognition, called NER, is one of the natural language processing tasks which involves the identification and classification of entities within text into predefined categories. Entities are defined as something with distinctive existence, and in our case, we want to detect the words belonging to gene symbols and protein names. Although there are existing datasets which contains the categories of genes and proteins, they are considering the genes and proteins, the two categories, as a same category, while we want to separate them into 2 categories. So we reconstructed the data set by separating the 2 categories from the existing data sets and combining them to each category we want to identify. After detecting the entities belonging to the categories in our interest, the relationship between the genes and proteins are inferred by relationship extraction called RE tasks. For training the models for the RE task, the words belonging to gene symbol and protein names are marked as the gene and protein with special character respectively. And there are 4 types of relationship between the genes and protein. So we label each type of relationship as one-to-one as single, enzyme complexes as and, isozyme as or, and no relationship as not. We manually created the data set, and we collected this amount of data. And in the last step, we processed the gene symbols and protein names by converting them into common IDs used the IDs commonly used in the biochemical databases, and organize them in a final form of GPR associations, like this way. Here are the results of training the models for each task. We optimized 3 types of the biomedical language models for each task by splitting the training and test data in an 80-20 ratio. And in case of named entity recognition task, BioBird showed the best performances among the three models for both type of categories. And for the relationship extraction task, PubMedBird showed the highest prediction performance. Regarding the structure of the model, the BioBird and PubMedBird they have some similar architecture, but the pre-training process is quite different. Like BioBird is inherited from the language model called BERT, which is trained by the Wikipedia corpus, and BioBird undergo the additional training based on the PubMed and PMC full text, while the PubMedBird constructed its corpus from PubMed and PMC the words from the PubMed and PMC, and they did the pre-training from the scratch by these PubMed and PMC articles. For BioGPT, it appears that the model was not adequately trained for the NER task because of the insufficient amount of training data. Since the BioGPT has more number of the parameters to be trained than the other two models. After training the models, we conducted a case study to construct the GPR association of E. coli by retrieving 10,000 articles from the published paper, and we collected the whole GPR associations from the articles. When comparing the collected GPR association with the existing metabolic network of E. coli, about 65% of them was matched perfectly, while about 20% of them were partial match. The predictions which did not totally match with existing one was most of them were the reactions with complex GPR association, like for example this 2-oxobutanoate formate lyase is catalyzed by the combination of isozymes and enzyme complexes, but we could only predict some of them. Yeah. And notably, we could find some new GPR associations which were not included in the existing data set. For example, these two reactions, they did not have the GPR associations in the biochemical database, but we could suggest these new GPR associations by applying our system. In summary, we have developed a new system for extract GPR associations by processing relevant articles from public databases, and we optimized the models for performing two natural language processing tasks. And for the datasets prepared in this study, BioBird showed the best prediction scores for NER task, while PubMedBird showed the best for RE task. And we expect that the result extracted from our system can expand the scope of the metabolic network of a cell, and therefore facilitating the study of metabolic engineering. I'd like to express special thanks to Min-ji Kim, who conducted the research together, and Professor Hyun-Wook Kim to provide the ideas and guide my research. And I'm grateful for my lab members and the funding agency who made this study possible. Thank you for your attention, and any comments and questions will be welcome. Thank you.
Thank you. Now open for questions and discussion. Okay, please.
Hello, thank you for the talk. I was wondering, so when you find one link between gene and protein, are you able to actually go back to the actual article, citation and sentences from where this interaction have been found in your model? Because I do understand that that's a problem with AI model when it's and GPT-based AI, where it mash up the data source, and so you cannot go back to the source. And I do recall a few years ago, before AI was a thing, there was software that I don't recall the name that was doing kind of old school data mining, and there was this this feature where you will have a gene-gene interaction that was mined from PubMed, but you could actually curate the data, and the expert could actually go back to this interaction and see the article, read the article, and verify that the software and the data mining did the valid interpretation.
So you mean that how can I retrieve the original paper?
The original source from where from which you extracted the gene-protein interaction.
Yeah. Thank you for the question. And we are doing this system. The system is processed by step. Like first, we collect the text of paper by the PMID or the PMC ID, and we separate the sentence or the paragraph, and we, like, from the sentences or the paragraph, we detect the entities, genes and proteins, and we can indicate where these entities come from. So yeah, so I think we can go reversely to find the origin of the interaction.
Great, thanks.
Okay, other questions? Or maybe from me. BioBart or BioGPT, aren't they evolving? What I mean is they are improving every day. So how can you keep the consistency, or how can you guarantee their results? When you add additional information, maybe the answer will change.
Yeah.
So how can you reproduce the same answer from those LLM?
I think if the model are developed in the new version, the system of optimization of the model are consistent, and we settle the system of optimization for each model. So if the model if a new version of model is introduced to us, we can directly adapt them to newly train the model to get the new version of the optimized system. And
So okay.
I'm not sure.
Yeah,
I'm not sure.
thank
Yeah,
you. So you need to write research papers quickly. Okay, before version up.